Part 7, Integrated System Recovery. Chapter 24, From Fragmentation to Coordination. Fragmentation develops when tissues operate under different structural conditions and no longer interpret shared inputs in the same way. Each organ continues to receive the same signals, substrates, and regulatory cues, but the membrane environment determines how those inputs are translated into output. When membrane composition diverges across tissues, responses become inconsistent in magnitude, timing, and duration. Coordination is not lost because signals are absent. It is lost because the system no longer shares a common structural framework for interpreting them. Restoring coordination requires convergence back to a uniform membrane baseline that enforces consistent behavior across tissues. Signal alignment is the first layer to return. Hormonal and neural inputs carry defined information, but their meaning depends on receptor environments within each tissue. When membrane properties stabilize, receptor clustering, activation thresholds, and termination kinetics begin to match across cells. The same input produces a similar response profile regardless of location. This reduces conflicting outputs where one tissue amplifies a signal while another dampens it, allowing signals to propagate through the system without distortion. Transport coordination follows as gradients become consistent. Movement of substrates, ions, and signaling molecules depends on controlled permeability and maintain differences in concentration. In a fragmented system, these properties vary, creating uneven distribution. Some tissues accumulate substrates, while others remain deprived despite adequate supply. As membranes rebuild, permeability decreases and gradients hold more effectively, allowing transport systems to deliver substrates according to demand. Distribution becomes proportional rather than opportunistic. Energy synchronization emerges as mitochondrial efficiency improves across tissues. When some cells operate under conditions of high leakage and others maintain stronger gradients, energy availability differs between compartments. This creates asynchronous behavior where processes accelerate in some tissues and slow in others. As membrane integrity converges, ADP production stabilizes system-wide, allowing processes that depend on energy to proceed at similar rates. This alignment reduces interference between pathways that previously operated out of phase. Temporal coordination becomes possible when signaling and energy supply operate within defined windows. Activation and termination of processes occur in sequence rather than overlapping unpredictably. Hormonal pulses produce effects that rise and fall within expected durations, neural signals propagate with consistent timing, and repair cycles complete without interruption. This sequencing depends on stable membrane conditions that allow signals to begin and end without distortion. Feedback systems regain coherence as outputs align with inputs. Regulatory loops depend on accurate representation of system state. When tissues respond inconsistently, feedback signals become unreliable, leading to overcorrection or persistent imbalance. With restored structural consistency, outputs reflect inputs more accurately, allowing feedback mechanisms to maintain equilibrium without excessive adjustment. Intersystem communication becomes more precise. Endocrine, neural, and immune systems exchange information continuously, but their ability to coordinate depends on consistent interpretation of signals. As membrane properties converge, these systems operate with shared rules, allowing signals to produce integrated responses rather than competing or conflicting effects. Remodeling cycles reinforce coordination over time. Each cycle replaces divergent structures with lipids that support consistent packing and signaling behavior. As this process continues, variability between tissues decreases, and the system approaches a unified structural state. Specialization remains, but it operates within a stable framework that supports coordination. Energy demand for maintaining balance decreases as fragmentation resolves. Fewer resources are required to compensate for inconsistencies in signaling and transport. This allows the system to allocate energy toward productive functions rather than continuous correction, further stabilizing coordination. From fragmentation to coordination, therefore, describes a transition from a system where tissues operate under conflicting structural conditions to one where they share a common membrane baseline. As this baseline is restored, signaling, transport, and energy production align, allowing the body to function as an integrated system with predictable and efficient responses to internal and external inputs. Reintegration of metabolic, immune, and neural systems. Metabolic, immune, and neural systems do not operate as independent modules. They share substrates, signals, and structural constraints, and their outputs depend on how consistently those constraints are maintained across tissues. Fragmentation separates their behavior by introducing variability at the membrane level, where each system interprets inputs differently. Reintegration occurs when this variability is reduced and a shared structural baseline is restored, allowing signals to be translated into coordinated responses rather than competing or conflicting actions. Metabolic signaling provides the substrate context for the other systems. Availability of glucose, fatty acids, and amino acids determines energy status and influences how cells respond to incoming signals. When membrane integrity is restored, transport systems deliver these substrates in proportion to demand, and mitochondrial efficiency converts them into ADP with less variability. This stabilizes the energy environment in which immune and neural processes operate, allowing them to function without compensating for fluctuating supply. Immune signaling depends on both substrate availability and membrane composition. In a stable system, inflammatory responses are initiated when required and resolved once the objective is achieved. When membrane conditions vary, immune cells interpret signals inconsistently, leading to either exaggerated or insufficient responses. As membrane properties converge, immune recognition, activation, and termination align more closely with input. This allows immune activity to integrate with metabolic state, responding proportionally to actual conditions rather than to distorted signals. Neural signaling requires precise timing and stable electrical conditions. Membrane integrity determines how well neurons maintain ion gradients and propagate electrical impulses. When permeability is controlled and energy supply is consistent, signal transmission becomes reliable. This allows neural outputs to reflect accurate information about internal and external conditions, coordinating both metabolic and immune responses through central and peripheral pathways. Cross-system communication becomes more coherent as these conditions stabilize. Hormonal signals that regulate metabolism influence immune activity, and immune mediators affect neural signaling. In a fragmented system, these interactions produce conflicting effects because each system interprets the same signals differently. With restored membrane consistency, these signals produce aligned responses, allowing systems to reinforce rather than disrupt one another. Feedback loops regain functionality within this integrated environment. Metabolic signals inform immune activity, immune responses influence neural perception, and neural outputs adjust metabolic processes. When outputs are consistent, feedback mechanisms operate with greater accuracy, maintaining balance without excessive oscillation or correction. Energy allocation becomes coordinated across systems. Rather than competing for limited or inconsistently available resources, metabolic, immune, and neural processes draw from a stable energy supply. This reduces the need for prioritization under stress and allows processes to proceed without disrupting one another. Temporal alignment improves as activation and termination occur within defined windows. Immune responses begin and resolve in sequence, neural signals propagate with predictable timing, and metabolic transitions occur without overlap or delay. This sequencing depends on consistent membrane conditions that allow each system to operate within shared temporal constraints. Structural remodeling reinforces integration over time. As membranes across tissues converge towards similar composition, differences in signal interpretation decrease, and system-wide coordination becomes the default state. This does not eliminate specialization, but ensures that specialized functions operate within a unified framework. Reintegration of metabolic, immune, and neural systems therefore emerges from restoring the structural conditions that govern how signals are processed. By aligning membrane properties, stabilizing energy production, and reducing variability in signaling environments, these systems begin to operate as interconnected components of a single network. Their outputs become coordinated because they are derived from the same underlying architecture, allowing the body to respond to inputs as an integrated whole rather than as a set of independent processes. Stability across variable conditions. A stable system is not defined by constant inputs. Is defined by the ability to maintain consistent output despite changing conditions. Temperature shifts, fluctuations in nutrient availability, variations in activity, and external stressors all alter the environment in which the body operates. Stability emerges when membranes provide a structure that absorbs these changes without allowing them to distort transport, signaling, or energy production. The membrane acts as a buffer, enforcing limits on how much internal conditions can fluctuate in response to external variation. Mechanical stability is the first requirement. Membranes must maintain their structure under changes in temperature and physical stress. Saturated lipids provide resistance to deformation, while monounsaturated lipids prevent excessive rigidity. Cholesterol adjusts local packing to maintain consistent behavior across a range of conditions. When this balance is present, the membrane does not shift unpredictably in response to environmental changes, and its permeability remains controlled. Transport processes depend on this consistency. Gradients that drive the movement of ions and substrates must be preserved regardless of external variation. When membrane integrity is stable, passive leakage remains low, and transporters operate under predictable conditions. This allows cells to maintain internal concentrations even when external levels fluctuate, preventing disruption of metabolic processes. Receptor function must also remain stable across conditions. Changes in temperature, substrate availability, or signaling intensity should not alter the fundamental behavior of receptors. A consistent membrane environment ensures that activation thresholds, clustering behavior, and termination kinetics remain within defined ranges. This allows signals to be interpreted accurately regardless of external variation. Mitochondrial membranes must preserve gradient integrity under changing demands. Increased activity raises energy requirements, while periods of rest reduce them. A stable inner membrane adjusts output by modulating electron transport without losing containment. Proton leakage remains low, allowing ADP production to scale with demand without introducing variability in efficiency. Oxidative resistance contributes to stability over time. Exposure to reactive species increases under stress conditions, but membranes that resist oxidation maintain their structure and function. This prevents the accumulation of damage that would otherwise alter permeability and signaling behavior, allowing the system to return to baseline after stress is removed. Temporal stability depends on the ability to initiate and terminate processes within consistent time frames. External variation should not extend or shorten these windows unpredictably. When membrane conditions are stable, signaling events begin and end according to internal regulation rather than external disturbance, preserving coordination across systems. System-wide coordination relies on shared stability across tissues. When membranes in different organs respond similarly to changing conditions, signals produce aligned effects. Hormonal responses remain proportional, neural signaling maintains timing, and immune activity stays within defined limits. This allows the body to adapt without losing coherence. Energy demand for maintaining stability decreases as structure improves. Fewer resources are required to correct deviations caused by external variation, allowing the system to allocate energy toward adaptation rather than continuous correction. Stability across variable conditions therefore arises from membranes that maintain consistent physical and electrical properties despite external change. By providing a controlled interface, they ensure that internal processes operate within defined limits, allowing the body to adapt to variation without losing precision, efficiency, or coordination. Consistent signaling across tissues. System-wide signaling depends on whether different tissues interpret the same input under the same structural conditions. Hormones, neurotransmitters, and immune mediators circulate or propagate with defined concentration and timing, but their effects are determined locally at the membrane. When membrane composition varies, receptors operate under different mechanical and electrical constraints, and identical signals produce divergent outputs. Consistency emerges when membranes across tissues converge towards similar packing, permeability, and domain organization, allowing signals to be translated into aligned responses. Receptor activation thresholds must match across tissues for signals to produce coordinated effects. Activation depends on controlled receptor mobility and the ability to cluster within organized domains. When membranes differ in fluidity or packing density, some receptors activate at lower concentrations while others require stronger input. This creates uneven responses where certain tissues amplify signals and others dampen them. As membrane properties stabilize, activation thresholds align, and the same signal produces a comparable level of engagement across cell types. Signal propagation relies on the assembly of downstream components within defined membrane regions. These interactions require consistent spatial organization so that signaling pathways proceed in a predictable sequence. When membrane domains are unstable or vary between tissues, the efficiency of propagation differs, leading to variability in signal strength. Rebuilding domain organization allows signaling complexes to form and function under similar conditions, producing outputs that reflect input rather than local structural differences. Termination of signaling must occur within similar timeframes to maintain coordination. Prolonged signaling in one tissue and rapid termination in another create temporal mismatch, where processes that should occur in sequence overlap or diverge. Membrane structure influences how quickly receptor complexes disassemble and how effectively baseline conditions are restored. When integrity is consistent, termination kinetics align, allowing signals to begin and end in synchrony across tissues. Transport of signaling molecules and ions must also operate under uniform conditions. Controlled permeability ensures that gradients are maintained and that movement occurs through regulated pathways. When permeability varies, distribution becomes uneven, altering local concentrations and affecting how signals are perceived. Stabilizing membrane structure reduces this variability, allowing signals to reach tissues with consistent intensity. Electrical conditions influence signal interpretation, particularly in excitable tissues. Membrane potential depends on maintained ion gradients and selective permeability. When these properties differ, the same signal can produce different electrical responses. Restoring gradient integrity aligns electrical behavior across tissues, supporting consistent signal transmission and response. Energy availability supports signaling processes at every stage. Activation, propagation, and termination require ADP. When mitochondrial efficiency improves across tissues, energy supply becomes consistent, reducing variability in signaling capacity. This ensures that differences in response are not driven by local energy limitations. Feedback systems depend on consistent outputs to regulate signaling. When tissues respond differently to the same input, feedback signals become distorted, leading to overcorrection or persistent imbalance. As responses align, feedback loops operate with greater accuracy, maintaining signaling within appropriate ranges. Remodeling cycles reinforce consistency over time. As stable lipids are incorporated across tissues, membrane properties converge, reducing variability in receptor environments and transport behavior. This gradual alignment produces a system where signals are interpreted under shared conditions. Consistent signaling across tissues therefore arises from uniform membrane structure that enforces common rules for activation, propagation, and termination. When these conditions are met, signals maintain their meaning as they move through the system, allowing coordinated responses that reflect input rather than local structural variability. System-wide synchronization. Synchronization emerges when multiple systems operate on the same timing, under the same structural constraints, and with access to a consistent energy supply. It is not imposed from a central controller alone. It arises when membranes across tissues enforce similar rules for signal interpretation, transport behavior, and electrical activity. When those rules match, independent processes begin to align in phase, and outputs across the body move together rather than drifting apart. Temporal alignment begins with signal initiation. Hormonal pulses, neural firing patterns, and immune activation events carry defined timing, but that timing is preserved only if receptor environments respond within the same window. Membrane composition determines how quickly receptors engage and how efficiently signaling complexes assemble. When this varies, identical signals arrive simultaneously but trigger responses at different speeds. As membranes stabilize, initiation kinetics converge, allowing signals to activate tissues in near synchrony. Propagation timing follows the same constraint. Signals must move through pathways at consistent rates to maintain sequence. This depends on domain organization, ion channel behavior, and maintained gradients. Variability in these properties causes delays or acceleration in specific tissues, disrupting sequence-dependent processes. Uniform membrane structure reduces this variability, allowing signals to propagate through networks with predictable timing. Termination defines the end of each signaling cycle. If signals persist longer in some tissues than others, processes overlap and interfere. Membrane integrity governs how quickly receptors disengage and how efficiently baseline conditions are restored. When termination kinetics align, signaling events close within the same time frame, preventing temporal drift between systems. Energy synchronization supports these processes. ADP production must scale with demand across tissues without introducing delays. When mitochondrial membranes differ in integrity, some cells meet demand quickly while others lag, creating asynchronous behavior. As gradient containment improves and electron leakage decreases, ADP availability becomes more uniform, allowing energy-dependent processes to proceed in parallel. Electrical synchronization is required for coordinated neural and muscular activity. Membrane potential depends on controlled ion gradients and permeability. When these are stable across tissues, electrical signals maintain consistent amplitude and timing. This allows coordinated firing patterns and reduces noise that would otherwise disrupt signal clarity. Substrate delivery must also align with timing demands. Transport systems distribute glucose, fatty acids, and other substrates according to gradients and transporter activity. When membrane permeability varies, delivery becomes uneven, causing some tissues to operate ahead of others. Stabilizing transport conditions ensures that substrates arrive in proportion to demand, supporting synchronized activity. Feedback loops enforce synchronization by adjusting outputs based on system state. These loops depend on accurate, consistent signals from all tissues. When outputs are aligned, feedback maintains timing across systems without excessive correction. When they are not, feedback amplifies variability. Structural consistency allows feedback to stabilize rather than destabilize timing. Circadian coordination integrates these layers into a repeating temporal framework. Hormonal rhythms, metabolic cycles, and neural activity follow daily patterns that depend on consistent responsiveness across tissues. Membrane stability ensures that these rhythms are expressed uniformly, preventing phase shifts between systems. Remodeling cycles progressively tighten synchronization. As membranes across tissues converge towards similar composition, variability in timing decreases. Processes that once operated out of phase begin to align, and system-wide behavior becomes predictable. System-wide synchronization therefore reflects the convergence of timing, structure, and energy across the body. When membranes enforce consistent conditions, signals initiate, propagate, and terminate within shared windows, energy supply matches demand across tissues, and feedback maintains alignment. The result is a system where independent processes operate in coordinated sequence, producing coherent and efficient responses to internal and external inputs.